All right, folks, let's get into our next macromolecule, which is proteins. Now, proteins are about 10 to 30% of the cell mass. They're going to include carbon, hydrogen, oxygen, nitrogen, sulfur, and even phosphorus. And they're so varied that I want you to make sure that you have a couple of proteins under your belt that you know of, and know their function. Okay, because there's just too many to choose from. There's structural proteins, which are there for mechanical support, like collagen. There's enzymes, which you get to talk about, like for catalysis or speeding up chemical reactions. There's transport proteins, which you get to talk about when it comes to transporting any type of enzymes or digestive enzymes, for example, between cells, okay, or even transporting oxygen in the blood. There's contractile proteins that we're going to talk about when we get to learn about muscle physiology. There's communication proteins when it comes to making signals across the actual body to make sure that these messengers can get their message to wherever they need to go. And then there's defensive proteins like antibodies that are released for immune systems. So because there's so many varied functions, you want to make sure that you have some proteins under your belt. Like with triglycerides, there's 3 of them. Carbohydrates, again, there's 3 different types. But for proteins, there's so many different types. I would say at least try to have 2 to 3 that you can always say, hey, I know what this type of protein is, and I know the overall function. Now, we get to focus on enzymes, so that might be easier for everyone to remember. But make sure you bring some other ones up there as well. All right, so we are back to monomers and polymers. So a monomer of a protein is going to be an amino acid, okay? And when a bunch of those monomers come together to form a polymer, it's now called a polypeptide. And some folks might say, well, where'd you get the peptide from if we went from amino acid to peptide? Well, the reason is when an amino acid bonds with another amino acid, they form what's called a peptide bond. And because they form a peptide bond, that's how we get the polypeptide, meaning many peptide bonds when it comes to the polymers of proteins. Okay, folks? Now, the beauty of these different amino acids is, well, the beauty of proteins is that they have different amino acids. Different amino acids means that they can have all the different functions that we saw before and different structures. But they all, every single amino acid does have something in common. So one thing they're going to have is an amine group. That's where the amino part comes from for the name. And an acid group. That's where the acid part comes from. So the acid part is going to be our carboxylic acid here. The amine group is here. And then it's also going to have an R group. And this R group is going to depend on whatever amino acid you're dealing with. And there's 20 types to focus on. Now, you don't have to remember any of the 20 types. When you get to biochemistry, you will. But for us, you do not have to remember that. Okay? So polypeptides, again, are going to be many amino acids formed because, or bonding together due to a peptide bond. It's still the same dehydration synthesis. It's just named differently because, again, we're focused on proteins. So, for example, I have amino acid here, I have amino acid here. I have my double arrow for my reaction. And if I'm losing water, then I'm going to create a peptide bond. As you can see here, I have a dipeptide, meaning I have 2 amino acids attached. Now, if I were to go in the reverse and I was adding water to this overall peptide bond, then I'll be breaking it apart and creating a decomposition reaction to make our 2 amino acids again. Same old that we saw before. Okay? Now, polypeptides are 10 or more amino acids bonded together in all those peptide bonds. But when you're talking about proteins are ginormous. Okay? So they could be 50 or more, or even 10,000 amino acids coming together. It just depends. And because we have so many different types of proteins, they all have different configurations that we're going to get into next. All right, folks, let's now get into levels of protein structure. So first, we want to realize that structure of a protein is going to dictate its function. And depending on the levels of its structure, this function will change. So for example, if you're at the first level, this protein is going to have a different function than the second level, which will have a different function in the third level, and of course the fourth level. So let's go ahead and start with the first level. First level is what we've seen before. We have a bunch of amino acids coming together to form peptide bonds. And so what we see here is a polypeptide. So that's kind of like the first level of protein structure. You can also say first level of protein folding as well, because a lot of times proteins are going to end up folding more and more as we go along. Now, the second level looks a little bit different, right? So what happens is one thing, and that is good old H bonding comes into play. Now remember, H bonds is going to be hydrogen attached to an oxygen, a nitrogen, or a fluorine. And for us, when it comes to secondary structure, then we definitely see that we have hydrogen and oxygens coming together. And one thing to remember that I don't know if I said, but I'll say it here, is that when you have a hydrogen bond, you usually see dots in between both of these elements. And so that's what you see in red here, little dots. That's going to signify, hey, I'm dealing with the hydrogen bond. And hydrogen bonding is going to happen because a lot of the polypeptides can start forming little hydrogen bonds in between one another to make it easier to form two different new structures at the secondary level. That's going to be our beta pleated sheets and our alpha helix or coil structure. So if the hydrogen bond is going this way, you can form like little coils. If they're going to be right next to one another, like maybe here, you can form sheets, and that is how you can get secondary structure. Also with third or tertiary structure or third level, you also have H bonding. Now the beauty of third level is that you finally get proteins that are 3D, okay? And we're talking about fully 3D. Yes, the alpha helixes can be looking 3D, but we're talking about a lot of different pieces, okay? So you can have your ribbons, you can have pleated sheets, anything that's going to help this protein have a specifically unique structure. As you can see, I see some alpha helices here, I see some polypeptide pieces here. All of it comes together to form a larger 3D structure, and that's what the third level is about. Then you go to the fourth level, which is basically taking two or more uniquely shaped proteins, like two or more of these three third or tertiary level structures, two or more of these, and these are now considered subunits. And when these subunits come together, they make these huge globular proteins, and that's where the quaternary structure or fourth level is going to come from. And so first level, second level, third or fourth, or primary, secondary, tertiary, or quaternary is going to be how these proteins can come with so many different functions, meaning the polypeptide is going to have a different function based on its structure. The beta pleated sheets and the alpha helixes coils are going to have different functions based on their structure. The 3D for the tertiary level is going to have a different function, and also the fourth level is going to have a different function, which makes sense since we have so many proteins that are so ubiquitous and have so many different functions in the actual body. Now, I did want to put a spotlight on enzymes, because I told you I was going to bring up one of them, and I'm going to choose enzymes because there's a lot more details with enzymes sometimes. And enzymes are those quaternary structure globular proteins. What they're going to do is act as a catalyst, okay? Now, what do we know about reactions before we even get into this? We know that if we have a chemical reaction, we have reactants going to products, or we have reactants going to products, and then the products going back to reactants if they're in equilibrium. That's what we've seen thus far in this lecture, right? So then we have to kind of apply energy to this in a different way. You know, we were talking about matter and energy. Now we're going to be applying energy to reactions. What we're saying is it takes energy to go from reactant to product. It takes energy to go from a product to reactant. How much energy is going to depend on the chemicals that you're bringing together to form brand new molecules. Okay? There's two types of, reactions that we can think of that take energy. You're going to have endergonic and exergonic reaction. What's the difference between the two? Well, first, let's go ahead and create some graphs. Boom, boom. We have reactants going to products here. We have energy. We have reactants going to products here, and we have energy. Now, with the endergonic, that means you're going to need energy in order to form products. You have to absorb energy to form these products. So you're going to be starting at a low energy for reactants, and then you're going to need to absorb energy in order to form products. Okay? Exergonic is the opposite. You're going to start off with the high energy for your reactants. Still going to need energy to go to products, but once you go to products, you're going to end up releasing a lot of energy. And that's what we can see here. Endergonic, you're starting off low, you have to absorb energy to get your products. Exergonic, you're starting off with high energy and reactants, and then as you go to your products, you're going to be releasing energy. And so because we have both of these endergonic and exergonic reactions, and every single chemical reaction has their own amount of energy associated with them, we can start tying this into our understanding of enzymes as we go along. How we're going to do that is by one little piece of this actual graph. This little hump here and this little hump here. These little tiny humps are going to be signifying what is called the activation energy. So activation energy. Activation energy is the amount of energy it takes for reactants to be converted into product, no matter what. Okay? And now you might be saying, okay, well, what do enzymes come into this? Well, the beauty of enzymes is that they're going to decrease this activation energy. So that means it's going to get your reactions to products way faster. So it'll make it go a little bit tinier hump down, a little tinier hump here. So you still need energy to get this process going, but to get from reaction to products, you can need less energy here due to an enzyme. And here's a better graph here. You have energy going to reactions to products. Again, to start off, we have our activation energy here. And this is without an enzyme. This is how much energy it's going to take to go to products. Now, if you're in industry or anything like that, sometimes you don't want your reactions to take that long. You want them to be quicker, faster, right? So you can get the product faster. That's where an enzyme comes in. An enzyme is going to allow us to decrease that activation energy, or that tiny little hump, just like we saw here. Okay, folks, now the question becomes, how does this protein do this? Well, first, it's going to act as a catalyst. And a catalyst is going to allow us to, again, decrease the activation energy, but not be used up in the process. So think about that. Oh, I can actually decrease my activation energy to allow me to get to the products faster, and I can continue to reuse this enzyme over and over again? Yes, that is what these enzymes do. And that is the beauty of this particular protein. It's going to allow for millions of reactions per minute. Now, how it happens, that's where we get a couple of different theories. The theory we see here is going to be the substrate active site theory. What that means is you're going to have this ginormous enzyme, which is what we see here. Now, the ginormous enzyme. And this enzyme is going to have a particular structure, just like we talked about before. And what do we say? Structure is going to dictate whatever the function is. Well, this enzyme is so specific that it has a specific site on it called an active site. And that active site is only made for particular substrates. And those substrates can be any reactants that we're talking about. So for us, we're talking about amino acids. So we have two of these amino acids coming together. Okay? Once you actually have the amino acids go into the active site, you create an enzyme substrate complex. So now we have our enzyme, we have our substrate here, and these two things are in the active site ready to continue on. And what do we see here? We see our arrow. Our arrow. So we're trying to go from reactions to products, and I'm showing you how enzymes do that. And once you have your enzyme substrate complex, then what's going to happen is a bond is going to be formed between these two amino acids. When a bond is formed, water is released because we're dealing with a synthesis bond, right? A synthesis type of chemical reaction. And now that that bond is complete, we have produced a peptide bond, which again is based on synthesis, a synthesis reaction, and we've just created a dipeptide. Now this dipeptide can go into another enzyme that has a particular active site for that dipeptide, and then it can keep on adding protein or amino acids to form different types of polypeptides. Now, as you learn and go higher, especially in biology, you'll realize that we do have, translation that happens where you can use ribosomes to help you build polypeptides as well. So there's a lot of different aspects of that. But once you have your dipeptide, then your enzyme goes back to just being an enzyme with an active site that's waiting for the next amino acid to come in. So think about that. Enzymes help speed up chemical reactions by decreasing the activation. And we have so many different types of proteins around. And we also have so many different chemical reactions in our body. So what does that mean for us? That means there's a bunch of enzymes in your body. Especially if they're so specific, you're going to need enzymes that's going to help with multiple combinations of these amino acids coming together. Because remember, the active site is super specific. It's only going to fit and do its job for those particular substrates. So that's the beauty of those enzymes in that way. All right, folks. Now let's go ahead and go back to our table here. We're now to proteins. Elements: carbon, hydrogen, oxygen, nitrogen, some sulfur. The building blocks for amino acids, that's its monomer. The function is going to be structure or functional in nature. Again, choose 2 to 3 different functions to write out here. Okay? And of course, write any note you want. That's just extra for you to remember. Why would this be helpful? Because as you go along and you're studying, you want to make sure that you have your own voice for this information and make it easier for you to answer questions. All right, folks, let's go ahead and move on to our last macromolecule.